Hi, Bio. Mrs. Cozen, and this is going to be video 3, DNA replication. I'm with my microphone here, so please let me know if when you guys are listening to this, you hear the sound going off at all, or if it's getting a little wonky. Let me know. I'm having a hard time hearing it from my phone or from my computer when it's not playing correctly on yours. So you guys let me know if it doesn't sound right. So DNA replication. Why do I want to copy this stuff? Why would I want to copy millions or billions of all these little base pairs? And the answer is when I need new cells, which you need new cells all the time. You need new cells to grow. You need new cells to repair when there's damage. You are probably making about 600 million of them right now. Okay. We're not exactly sure how many at any point in time, but you're probably making a lot of cells right now. So you're probably doing this. You're not probably, you are doing this right now, millions and billions of times. So to copy DNA, let me kind of walk you guys through the process first. Then I'll give you the notes for it. Then we'll watch a quick video clip about it. So first thing is double helix. I want to copy this whole thing, but I don't want to just kind of go off to the side and make a whole copy. I want to use what I have as a template to make a new strand of DNA. So the first thing that I do is I unzip right here these hydrogen bonds. Okay, so the hydrogen bond's not super strong. They're strong enough to hold our DNA together, but we have an enzyme called helicase that is going to unzip them just like the zipper on your coat. Zoop, zoop. Okay, we're going to break those up. And so then what we're going to end up with is our DNA, which was connected with our hydrogen bonds. It's now split open. Think of it like teeth on a comb or something like that. Okay, so we've got my A and it was matched up with a T, and a G was matched up with a C, and maybe another T and an A. Okay, so when I bring those out, I can now use each side as a mold or a template to build a new one. We can follow the rules of base pairing. And now let's say this first one was a T. Okay, so when I unzip these, okay, and this one was a T, now I can bring in a brand new A to pair with that T. Okay, and I can bring a brand new C to pair with that G until I get enough so that I can make myself a new half to that double helix. Now, when would this happen? This would happen in mitosis or meiosis during that interphase. Remember when I gave you guys the clock and I said, if you looked from 12 to 9, if it took you an hour to make a new cell, which it probably takes you longer than that, if it took you an hour to make a new cell, 45 minutes of that hour are spent checking, double checking, and copying. And that's this process right here. So this process of DNA replication, it happens in a lot of different places. There's a lot of proofreading to make sure that everything's right. Okay, but that takes up a huge majority of the cell cycle. And that last part was the prophase, metaphase, anaphase, telophase, cytokinesis. Okay, that was the last part. But this part right here, interphase, when we're replicating and checking, that's where this DNA replication happens. So the first thing is it has to happen in hundreds of places at the same time. If we just tried to go all the way down your strand of DNA from one side to the next, it would take something like 11 years to copy the DNA just in one cell. And we don't have 11 years. We need to be growing right now. So it happens in hundreds of places at one time. And it also happens in both directions. So remember, you guys, I said we take that original DNA and we split it in half. Okay. We have hundreds of places where the matching is happening, but it's also happening in both directions. Okay, so it actually ends up occurring, if you think of each side as one section, it's happening kind of in thousands of places at a time. So it's happening in a lot of different places and it's happening in opposite directions. When we are pairing up DNA, we may have talked already about the idea of anti-parallel. That anti-parallel idea is really important, okay? You have DNA that's working in two different directions, and also replication works in two different directions. Okay, so the very first step, I've already talked about this, it's where we separate the DNA, we unzip it with the enzyme helicase. I want you guys to make sure that helicase is either underlined or highlighted. You need to make sure that's the enzyme that's going to separate our DNA. Okay, then, so enzyme comes in, enzyme helicase unzips it, and we have two halves now. And now we have a little enzyme called DNA polymerase, and it's going to come in and it's going to match up the base pairs so that we can get half old DNA and half new. Okay, and then over here we had our side, DNA polymerase comes in, matches up the base pairs. And remember, that's happening in both places at once, but I don't have 4 hands, so I can't show you that all at once. Okay, next step. Now our double helix, the new one that we have, is made out of half old and half new, and we call that semi-conservative. It's really important because it saves our cells a lot of time and a lot of energy. If we just had to like build a whole new DNA over here, it would take way too much time. So we use what we have as a model. All right, let's watch a quick little replication video so you don't have to keep looking at my hands as base pairs.
A double strand of DNA unwinds, and the strands separate.
All right, guys, that was helicase that unwound the DNA, okay? So now we have half of our old DNA, and you can see it's kind of open, and it'll be ready now for the correct base pairs to come in.
For each unwound strand, the bases can now match with those that are floating free in the cell.
And the thing that's doing this is called DNA polymerase. And DNA polymerase has two jobs. Number one, it's going to help to match up the base pairs. The other thing it's going to do is it's going to proofread and make sure that everything's right, and it can go back and make corrections.
The familiar base pairing rules apply. A pairs with T, G with C, and so on. So each strand of the double helix acts as a template for the formation of a new complementary strand. This eventually gives two DNA double helices, each identical to the original and actually containing one old strand and one new.
All right, so that's it, you guys. That's how DNA replication works, and we're going to do a lot more in class about this, okay? One more picture to show you, just another representation of replication. Remember, I want to show you as many models as I can throughout this unit. In this one, you guys have your DNA coming in. It's in on both sides. The spot where it's open, helicase has opened that. And then these little things that are they look like little green balloons. You can see an arrow pointing this way. This is DNA polymerase. And this first one is working in this direction, and the other one's working to replicate in the other direction. So at the end, we'll end up with one whole strand of DNA here and another one over here. All right, your summary. So you guys are going to use the rules of base pairing a lot here. Remember that T goes with A and G goes with C. Write the questions, write the answers. Okay, half of my original DNA is made up of T, A, G, A, T. Okay, so that's half. So it's split, and think of that as this half. Now I want to know what's the complementary strand. So complementary strand could either mean the old strand that I separated it from, or it could mean the new strand that I'm connecting it to. It'll be the same thing if you follow the rules of base pairing, okay? So for example, T goes with what letter? A. So A would be the first letter in the complementary strand. Okay, second one, two enzymes we use in replication. Third one, very similar to number one. If my new DNA that I replicated is GATTAC, okay, what was the original strand that I used as a template? Okay, so example, G goes with C. That's what you would start the original strand with. All right, so you guys, that's the end of video 3, replication.